Immunosorbent Assay. ELISA is a plate-based assay technique designed to detect and quantify substances such as peptides, proteins, antibodies, and hormones. The fundamental principle of ELISA involves 4 key steps. First, antigens are immobilized to a solid surface, typically a microplate well. Next, antibodies specifically bind to the target antigens. These antibodies are linked to enzymes such as horseradish peroxidase or alkaline phosphatase. When the enzyme substrate is added, it produces a measurable product, often a color change that can be detected. There are 4 main types of ELISA assays, each with unique configurations. Let's examine each type in detail. In direct ELISA, a primary antibody directly linked to an enzyme binds to the immobilized antigen. Indirect ELISA uses a primary antibody that binds to the antigen, followed by an enzyme-linked secondary antibody. In sandwich ELISA, the antigen is sandwiched between a capture antibody and an enzyme-linked detection antibody. Competitive ELISA involves sample antigens competing with reference antigens for binding to antibodies. ELISA uses various detection methods to amplify and measure signals. Colorimetric detection involves a visible color change with intensity proportional to the analyte concentration. Fluorescent detection uses fluorophores that emit light at specific wavelengths when excited. Chemiluminescent detection captures light produced directly by a chemical reaction, offering high sensitivity. ELISA is characterized by its exceptional sensitivity. This high sensitivity makes ELISA invaluable in clinical diagnostics and research applications. ELISA can detect molecules at concentrations as low as picograms per milliliter, making it one of the most sensitive immunoassay techniques available. Enzyme-Linked Immunosorbent Assay, or ELISA, has several critical applications in immunology. ELISA serves as an essential tool across different areas of immunological research and diagnostics. It's widely used in disease diagnosis, particularly for conditions like HIV, where antibody detection is crucial for identification. In vaccine development, ELISA helps researchers monitor antibody responses and test immunogenicity of vaccine candidates. It's crucial for measuring immune responses, including cytokine levels in inflammatory conditions. And ELISA is valuable in allergy testing, where it can detect allergen-specific IgE antibodies in patient serum. The ELISA procedure follows several critical steps that ensure accurate and reliable results. First, the microplate wells are coated with a capture antibody that specifically binds to the target molecule. Next, nonspecific binding sites are blocked with proteins like bovine serum albumin or BSA. The sample containing the target molecule is then added to the wells. Detection antibodies conjugated with enzymes are introduced, which bind to the captured target molecules. A substrate is added, which is converted by the enzyme into a colored product, producing a measurable signal. Finally, the results are read with a spectrophotometer, which measures the intensity of the colored product. ELISA is widely applied in various specific diagnostic and research contexts. In HIV diagnosis, ELISA is the primary screening test detecting HIV-specific antibodies in patient serum with high sensitivity. For inflammatory conditions, ELISA can precisely measure cytokine levels, providing valuable insights into disease progression. In autoimmune disease detection, it identifies autoantibodies that target the body's own tissues. And for allergy testing, ELISA measures allergen-specific IgE antibodies, helping diagnose and manage allergic conditions. ELISA offers several important advantages while requiring careful optimization. Its advantages include high throughput capability, allowing testing of many samples simultaneously, and providing quantitative results that can be precisely measured. However, ELISA requires careful optimization of several parameters to ensure accuracy and reproducibility. ELISA's wide range of applications and well-established procedure make it an invaluable tool in immunological research and clinical diagnostics.